Enzymes enable molecules called substrates to undergo a chemical change to form new substances called products. Each enzyme acts on a specific molecule or set of molecules called substrates. Each substrate fits into an area of the enzyme called the active site. This fitting together is often compared to a lock and key mechanism. However, the enzyme changes shape a little to fit with the substrate. In the enzyme-substrate complex, the enzyme holds the substrate or substrates in a position where a reaction can occur easily. After reaction, the enzyme releases the products and can go on to carry out the same reaction again and again.
Here's an example of a chemical reaction. One large compound is broken into two smaller compounds. This reaction takes place much too slowly to be useful in a cell. In order to speed up chemical reactions, cells make special proteins called enzymes. An enzymatic reaction begins when a specific compound binds to an area on the enzyme called the active site. The compound that binds is called the enzyme's substrate. Notice the shape of the substrate and the active site. They fit each other like a lock and key. In general, each enzyme's active site can fit only one kind of substrate, and so can speed up only one kind of reaction. When a substrate binds to the active site, an enzyme-substrate complex is formed. A slight change in the shape of the substrate weakens a chemical bond. This weak bond is easily broken, splitting the substrate into two pieces. The pieces, now referred to as products, detach from the enzyme. In enzymatic reactions, only the substrate is changed. The enzyme itself is unaffected by the reaction. After the products leave the active site, the enzyme is free to speed up another reaction.
Lock and key theory of enzyme action. Here, the larger enzyme molecule comes in contact with smaller substrate molecules to form an intermediate complex. This intermediate complex or enzyme-substrate complex soon breaks to release the enzyme and product of biochemical reaction. Mode of enzyme action. Biosynthetic reaction. Here, the active sites establish contact with substrate molecules to form products of biochemical reaction. Mode of enzyme action. The enzymes bear distinct cavities specially designed for only specific substrate molecules with proper geometric shapes that can fit correctly.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
Usually, every time you have a cut or bruise, your blood clots to stop the bleeding. Clotting is a complex process, and there are four main things that make it possible: platelets, or cell fragments; clotting factors, which are special proteins; fibrin, which is a protein mesh made up of a special kind of clotting factor;
and finally, other cells, like red and white blood cells. Because clot formation is so important, platelets and clotting factors are always available, floating around in your blood. Now, let's talk about the 4 steps involved with forming a blood clot. When there is a tear in a blood vessel, the first thing that happens is that the nearby platelets are activated and become sticky. They start sticking to each other and to the sides of the hole in the blood vessel. For small holes, enough platelets usually stick together to form a temporary plug. But a platelet plug isn't strong enough to block the opening for long, so it must be reinforced with other materials. Otherwise, the blood flowing past the hole could wash the plug away. These other materials are called clotting factors. As the clotting factors float by the tear, they become activated or turned on and add themselves to the platelet plug. A special kind of clotting factor can weave itself together with others of the same kind and form a web of fibrous tissue, called fibrin. This web acts like glue and holds the platelets and the other clotting factors together, creating a blood clot. Other cells, like red and white blood cells, can also get caught in this web and help reinforce the clot. It quickly becomes stronger and starts to pull the edges of the hole in the blood vessel together. After the clot has formed, it stays in the hole until the tissue is repaired. When the clot is no longer needed, your body dissolves it.
Cyclic AMP is a signaling molecule in biological systems. It is produced by adenylate cyclase, an enzyme activated by a G protein during cellular signaling. Because it can interact directly with kinase enzymes, it represents an advanced stage of cellular signaling. Typically only a few steps away from the final cellular response is carried out by a phosphorylated protein. The means by which CAMP turns on kinases has been extensively studied and represents a nice example of how substrate binding can remarkably alter the conformation of a protein. Before discussing how CAMP propagates cellular signals, we need to know something about the protein that it acts on, protein kinase A, or pKa. pKa is a member of a general class of proteins that phosphorylates, or adds a phosphate group to, residues on other proteins. Typically, this leads to activation of the phosphorylated protein and the execution of some chemical reaction or structural response. Phosphorylation, therefore, represents a method by which cells can turn on chemical reactions rapidly and controllably. Kinase enzymes are the biomolecules that control cellular responses due to phosphorylation. What then controls kinase enzymes? In the absence of cyclic AMP, pKa is present in the cell in an inactive tetrameric form consisting of 2 catalytic, or C subunits, and 2 regulatory, or R subunits. Residues on the R subunits normally block the active sites of the C chains, and CAMP is needed to activate the kinase enzyme for phosphorylation. In the presence of high concentrations of CAMP, as during the propagation of an extracellular hormone signal, the catalytic subunits are released from the regulatory subunits, and pKa can carry out its function of phosphorylation. CAMP thus acts as a gatekeeper to control the activity of protein kinase A. Detailed studies of the structure of pKa in its inactive form have been carried out. What you're looking at is a crystal structure of an R subunit complexed with a sulfur-containing analog of CAMP, showing the interactions that lead to CAMP binding to the regulatory subunit. As you can see here, the highly conserved phosphate binding cassette, or PBC, binds the phosphodiester portion of this CAMP analog through hydrogen bonds to arginine 209 and alanine 202, and the 2' hydroxyl is hydrogen bound both to glycine 199 and glutamate 200. Interestingly, the 2' hydroxyl is acting both as a hydrogen bond donor and acceptor in the PBC. Moving to the other side of the regulatory subunit, we see the same set of residues in a second CAMP binding domain. Hydrophobic interactions, including a key pi stacking interaction with an adenine capping residue, tyrosine 371, serve to bind the heterocyclic portion of CAMP. In fact, mutation of these residues resulted in the need for substantially more CAMP in order to activate pKa, supporting their critical role. Here you can see the structure of an inactive pKa complex containing an R and C subunit. The two CAMP binding domains of the R subunit are highlighted in blue for domain A and orange for domain B. Notice that the key adenine capping residue for domain A, ch15, is now 30 angstroms away from the phosphate binding cassette. CAMP binds only very weakly to domain A in this conformation, and a key ionic interaction or salt bridge between ARG366 and Glu261 holds the R subunit in this conformation. Previous kinetic data had suggested, and this observation confirms, that CAMP binding to the R subunit takes place in a stepwise fashion. CAMP binds in domain B, a conformational change in the R subunit occurs, then a second molecule of CAMP can bind in a much improved binding site in domain A. Because CAMP binding in domain B pulls the adenine capping residue tier 371 towards the PBC, it likely disrupts the salt bridge, allowing domain A's adenine capping residue to migrate towards its PBC. Binding of CAMP to domain A likely then induces a second conformational change, which leads to release of the C subunit for catalysis. Returning to the crystal structure of the CAMP-bound R subunit, we see that a substantial crimping of the long alpha helix in domain B is present, bringing the two CAMP binding domains into close proximity. This may also pull the N-terminal domain that binds the C subunit, not shown, in towards the binding domains, releasing the C subunit for catalysis. Unfortunately, the mechanism of C subunit release has not been studied in detail.